Critical headlines regularly promise revolutionary medical breakthroughs. Recently, many have seen claims about something called the XO001 targeted exosome platform, with reports suggesting it can completely eradicate 4 major cancers without long-term recurrence. It sounds like a miracle, a finish line for cancer treatment.
It certainly captures attention, and for patients and their families, those words are incredibly powerful. We're talking about research from China Medical University Hospital in Taichung, Taiwan. The work itself is ingenious and represents exciting scientific advancement. But as you suggest, we need to distinguish between a scientific milestone and a clinical reality. We have to objectively look past the adjectives and into the data. Are we looking at a medical finish line or a promising starting block?
That's exactly it. The most compelling claim, the 100% clearance of cancer cells, is often followed by a small but crucial phrase, quote, in multiple solid tumor animal models, end quote. This mice versus men gap seems to be a significant information challenge in medical journalism.
It absolutely is. Laboratory mice are the foundation of oncology research, but they are simplified models. They are often immunodeficient or inbred, which allows human tumors to grow consistently for study. Humans, by contrast, are genetically diverse, with complex environmental exposures and immune systems trained over decades. There's a somber joke in science, quote, we have cured cancer in mice for 40 years, end quote. The leap from a 25-gram rodent to a 70-kilogram human involves massive changes in drug metabolism and can reveal unforeseen toxicities.
So even if the treatment works perfectly in a mouse, that's no guarantee for a person. And what about the concept of cure in these studies? How does the timeline differ?
That's another critical point. In a mouse study, long-term survival might mean 60 to 90 days. In human oncology, we are very careful with the word cure. We talk about 5-year survival rates. A headline claiming no recurrence based on just a few months of mouse data is, at best, premature, and at worst, it distorts clinical terminology.
This XO001 platform is described as an evolution of CAR T-cell therapy. Could you explain what traditional CAR T involves and how XO001 aims to improve upon it?
Certainly. Standard CAR T therapy has been revolutionary for blood cancers like leukemia, but it's essentially personalized manufacturing. It requires extracting a patient's T cells, genetically engineering them using a viral vector in a specialized clean room, then expanding billions of these engineered cells before reinfusing them. This process can take 3 to 4 weeks and cost upwards of $400,000, which is often too long and too expensive for patients with aggressive, fast-moving cancers.
And the XO001 platform aims to streamline that complex process?
Yes, it attempts to turn the patient's own body into the laboratory. Exosomes are tiny bubbles, nanovesicles, secreted by cells for communication, like the mail trucks of the biological world. Researchers load these exosomes with genetic instructions for the CAR, which acts like a GPS, and bytes, which are bispecific T-cell engagers. When injected, these engineered exosomes are supposed to specifically find T-cells in the patient's spleen or blood, hand off the genetic package, and transform the T-cells into cancer killers directly inside the patient's body.
That's an elegant solution if it works. It would eliminate the need for expensive external lab procedures and potentially reduce costs drastically, allowing for off-the-shelf treatment. But what are the scientific hurdles this approach still faces?
The main challenges are whether these exosomes can find T cells in sufficient numbers without being quickly cleared by the liver or kidneys first, and if they can effectively alter T cells once they reach them. These are precisely the questions that clinical trials must answer.
The report specifically mentions lung, pancreatic, colorectal, and glioblastoma cancers. These are solid tumors, which are notoriously difficult to treat with immunotherapies. Why is that?
Solid tumors present a formidable barrier. Unlike liquid blood cancers, where cells float freely, solid tumors build what's called a tumor microenvironment, a literal fortress. There's a physical barrier, a dense stroma, or scar-like tissue that prevents T cells from even entering. Then there's a chemical barrier. Tumors secrete acid and exhaust oxygen, creating a dead zone where immune cells become exhausted and die. Finally, there's an invisibility cloak, where tumors express checkpoints like PD-L1 that tell T cells, I'm a normal cell, don't attack me. So while EXO-01 showed success in mice, the human TME is far more hostile. The claim of completely clearing these cancers in humans is currently a hypothesis that hasn't met the reality of human biology.
This brings us to a major red flag in the reporting, the juxtaposition of technology transfer completed and starting trials next year. In medical research, this sounds like what's known as the Valley of Death.
It is. The Valley of Death is the significant gap between a promising lab discovery and a commercial drug. An anti-cancer drug must survive 3 phases of clinical trials before it can be approved by regulatory bodies like the FDA. Phase 1 tests safety on a small group, and many miracle drugs fail here due to unforeseen toxicities in humans. Phase 2 assesses efficacy on a larger group to see if tumors shrink. Phase 3 compares it against the current standard treatment on an even larger patient population.
And the success rate through all those phases can't be very high?
Statistically, only 9.6% of drugs that enter phase 1 ever make it to FDA approval. When a headline declares a cancer wiped out before phase 1 has even begun, it's betting on a 90% chance of failure as if it were 100% certainty of success.
So, for our listeners trying to navigate this landscape of health news, what are some reliable filters they can use to protect themselves from hope peddling?
There are 4 key filters. First, the subject filter. If the article doesn't mention human clinical trials, treat it as a science experiment, not a medical treatment. Second, the peer review filter. Was this published in a reputable peer-reviewed journal, like Nature or The Lancet? The EXO-001 study was accepted by advanced science, which gives it scientific credibility, but the interpretation in the news is often where the clickbait lies.
And the other 2.
Third, the mechanism filter. Does it explain how it works, or does it just use vague terms like breakthrough and miracle? A good article provides some scientific detail. Finally, the expert filter. Look for quotes from independent doctors not involved in the study. If the only person quoted is the CEO of the biotech company or the lead inventor, be wary of potential vested interests.
That's a very clear framework. While we are in a golden age of medical advancement and technologies like EXO-001 are genuinely exciting, science moves in increments, not giant leaps. Declaring a cancer wiped out prematurely really overlooks the rigorous decade-long process required to ensure a drug is safe and effective for patients.
Exactly. We should remain hopeful, but our hope needs to be anchored firmly in evidence. The best medicine truly is a well-informed mind. Support the researchers, follow the trials, but never let a headline override your critical thinking.
If you found this discussion insightful, consider sharing it with someone who might also benefit from a more critical look at health news.